Hi everyone, welcome to Genomify. Today we're going to be talking about the technique called differential expression analysis. This is a technique applied to RNA-seq datasets. Applications can include transcriptional changes after drug treatment or looking at transcriptional changes after a gene knockdown. But today we'll be using DESeq2 to perform this analysis, and the data that we will be using comes from a paper I was a co-author on that was published about a year ago. But in this paper, we had taken a Wilms tumor patient-derived cell line and treated it with either a negative controlled DMSO or the experimental condition, the nuclear export inhibitor called KPT330. And we wanted to look at which genes were being upregulated or downregulated in response to this particular drug. And our goal was to identify genes that were differentially expressed upon treatment with this nuclear export inhibitor. And ultimately, we identified this gene called TRP13, which we showed was downregulated in response to this drug. Before we get started, our version I'll be using today is 4.4.2, and these are the 6 packages that need to be installed before you get started with this analysis. All right, well, let's get started. So today we're going to be covering a relatively basic bioinformatics technique called differential expression analysis, which is the technique applied to RNA-seq data when you're trying to see which gene transcripts are being upregulated or downregulated. And in this case, we're going to be using the package called DESeq2. And so to begin this analysis, you need to first read in the packages necessary for R to do all the things it needs to do. And here, I've run them all in. This is assuming they're already installed. And so then next, what we need to do is read in the metadata, which is providing information for each of the quantification files. I'm assuming you already have your quantification files. I will be releasing another video soon explaining how to actually perform this gene quantification, and that will then flow into this analysis here. But for now, we're going to assume you have these quant files. And so when you read in this metadata, this just tells you the name of the quant file, the cell line it's associated with, in this case just one, and the treatment, either the controlled DMSO or the experimental condition where we treated with a nuclear export inhibitor, KPT330. And then next, what we need to do is read in the associated transcript to ID metadata. This is essentially just telling downstream packages how to collapse multiple isoforms onto one gene ID. Each gene has multiple transcripts associated with it, so in this case we will just be collapsing them all to the 20 or so thousand known protein coding genes in the human genome. So here we're just reading in the transcript to gene ID metadata, which we're using a tool called BioMart, and so this will pull data from a server. In this case, you can see actually sometimes this can be an unresponsive request. This happens pretty frequently, so if you're having an issue, just keep trying it over and over or refreshing your RStudio. Looks like we were able to read it in this time, but if you're having issues with this, try restarting your R and it should typically work. And so then now we need to filter this object for protein coding genes only. In this case, we perform poly-A tail enriched RNA sequencing analysis, so we're going to be only keeping the transcripts that are associated with protein coding genes. And we're also, instead of using the ENSG identifier for each gene,
We're just going to be using the more classical external gene name, for instance TP53, or another more famous gene that you may be thinking of. But we don't really like to keep the ensemble IDs, as those are hard to interpret. So we're going to convert all of these to a kind of more traditional gene name. And here is where we are filtering for only protein coding genes. Alright. And so here we're performing all this filtering. And when you do all this, you will get this data frame that has the ENST or transcript ID and the associated external gene names. As you can see, there are a lot of transcripts. There are actually only around 20,000 in the human genome, so you'll see that later down the line. So once we've done this, we need to then actually read in our quantification files into R using this package called TXimport. And so first we need to create this nested list that will tell TXimport where are these quant files. And so here I'm just providing the path. I put them in this folder called quants in the directory I'm working in. And I'll show you what this looks like once I'm done. But this is the format that TXimport is expecting, and it will let the package know exactly where all your files are. So if we run this, you can see we get this nested list providing the file name and the path in which you can find that file. So now that we have that, we then want to read in our quantification data with this TXimport package, providing that nested list file there. We're telling it how to perform the quantification. In this case, we use salmon as the quantification method. Here we are providing the transcript to gene ID data frame, which we just created in our previous code chunk. And now we're just telling TXimport what are the file name or the column names in this quantification files we're providing. And so these are the associated column names in the quantification files we're reading in. And then we have these options that you have to provide. I've found that when you're doing this TX to gene conversion, TXimport just requires these to be true, and it will work, so I wouldn't worry about it too much. Just trust the process. And so now you can see that TXimport is reading in the 4 quantification files associated with this particular experiment. And then now we have it as a TXimport data format. We need to convert it to a DESeq format so we can perform differential expression analysis. And so this is now using DESeq2 to actually convert this TX data format to a DESeq2 format. And this will allow us to perform differential expression analysis in subsequent steps. And so this design here is telling DESeq2 what do you actually want to compare. In this case, we're comparing control to drug treatment. This is basically just dependent on the column names in your metadata file. And so now we have a DESeq2 object here. You can see it in our environment. Last, we need to perform one final filtering step. We like to keep only genes which have at least 10 counts across all conditions, so we're not comparing very tiny changes in gene expression. So we're going to perform this masking technique where we keep only rows with greater than 10 counts, and then we're going to filter this DESeq2 object. to only keep rows with greater than 10. Okay, we're back. Looks like this was somewhat of a memory-intensive process, and given I was screen recording, I had to shut down some other windows. But keep that in mind if you have an older laptop like me. This may cause some issues, but nonetheless, we have filtered for rows that have less than 10 total counts for a gene. And so then next, I'd like to perform some basic QC just to make sure the data we're looking at makes sense. So the first thing I do with RNA-seq analysis is a quick principal component analysis plot just to make sure the samples are grouping like we would expect. In this case, we want DMSO to cluster together and the drug-treated cells to cluster together. So the first thing we need to do to actually perform this analysis is create a normalized counts matrix, which is what will actually be used to perform the principal component analysis. And we're really using a normalization part of DESeq2 called VST. And then we'll convert this to a transposed object using the dplyr package. Not sure. Let's double check. And so this gives us a very large data frame where you have the samples here and the gene IDs up top there. And then we performed principal component analysis with a base R package called prcomp. And I just tell prcomp where are the normalized counts and put this into a PCA object. And here I'm just converting this object to a data frame, so it's easier to plot using ggplot2. Now we'll actually be plotting the top two principal components. But first we need to overlay the metadata on top of our PCA analysis. And so the way we're going to do that is tell the PCA samples object to combine with the metadata file, essentially. And the way we'll be doing that is Tibble and using the matched file names in each data frame to essentially join the two data frames together. Here we're just telling that there's this other data frame with matching values in the file name column that we want to join these rows together. And this will, I must have made a capital A. This will make sure your column names are correct. And so then now this pulls in the metadata into this PCA file. And here we have the principal components. We'll be plotting the top two. And this will essentially just show us how globally these samples compare. And so here's just a quick ggplot command that will show us if the samples are grouping like they should. X equals to PC1, Y equals to PC2. And we want to color by treatment so we know which circle belongs to which treatment category. And we just want to make the points a little bigger so we can see them on the screen. Let's see, this should work. And now if we look at this plot, we can see that the DMSO samples are clustering together, and the drug-treated samples are clustering together. I'll likely make another video on PCA plots later in the line, but this is just a quick and dirty QC metric just to make sure everything is as you expect. And so now that we know everything looks good, I want to actually start performing differential expression analysis. So first, we want to set the factor level so we know what is the numerator in the differential expression analysis. You can compare in opposite directions. So we want to make sure the DMSO is the denominator in this case. So everything is relative to the DMSO control. And so the way we do this is just re-level the treatment column in this DESeq2 object. So we're basically telling DESeq2 this is what you want to be the denominator in this analysis. And now that you've done that, you can actually perform the differential expression analysis on the DESeq2 object. Not the normalized. You don't want to perform differential expression analysis on the normalized object. Only unnormalized goes into this. And so now we have that. We'll then read that into the results, which converts it into a more digestible data frame that we can read. And I'll show you what that looks like once we're done here. And we'll just convert that to a data frame so we can look at it in R, because you can't really look at the DESeq2 objects by clicking on them in the environment. And so if we come and look at the results of the differential expression analysis, we have in the column, or sorry, row names, the gene name, and we have these associated values for each gene. In this case, base mean just looks at the overall expression between both conditions in general. Here we have log-fold change, which is really one of the most important metrics we're going to be looking at. And I won't dig into these too much, perhaps in a later video, but really the two columns we'll want to be looking at are log-fold change and p-adjust. And we want this to be below 0.05, which has corrected for multiple testing. And log-fold change, we tend to use -2 or +2 in my lab, but you can also use -1 and 1 depending on your analysis. And so as most people want to do, they want to export this data so they can actually start looking at it. So we were going to want to filter for genes that are either upregulated or downregulated. And the way that we define that, like I just said, an upregulated gene has a p-adjust value less than 0.05 and a log-fold change of greater than 2. So we want to filter for rows that meet those criteria. And so we'll use dplyr to do that. And so we want to use an AND command to make sure they meet both of these criteria. And that'll give us the data frame with only the upregulated genes that are significant. And we similarly want to do something for the downregulated genes as well. It's going to be the same thing, but log-fold change will be negative as opposed to positive. Make sure it's less than -2, not greater. All right, and then we will combine these to make one file we can export. A lot of times colleagues will ask me for a list of significant genes, and I tend to export that in Excel file format so that it's easily accessible to anyone not familiar with R. And last, we just need to change the row names to column names to a column, because this export function does not include the row names. Kind of a technicality, but nonetheless we have to do it. And we'll name the row name column gene. you can see this is what our exported file should look like in Excel, and so we'll use the right Excel XLSX, provide the file we're exporting, and we'll tell it where to save this, which is in the folder I'm working in. And so if we just run all that together, I did not import the right Excel package above. So let me do that really quick, and then do this, and that should work. And that we can look at later. And so then lastly, we will just perform some basic exploratory of the results. Make sure this matches what we expect. This is looking at a p-adjusted of 0.01, so we're seeing a few more, but yeah, this matches what you would expect here. And then lastly, we're going to want to plot the data. This is a relatively crude way of plotting differential expression data, and I'll have a video coming down the line that'll show more detailed ways to do this, but this is really just to show you that your differential expression analysis worked, and you're seeing genes that are being upregulated as well as downregulated. This is showing the significant genes in blue and the non-significant genes in gray, but we'll have to log transform this down the line to actually make a volcano plot that you may have seen in a couple other papers. But with that, congrats. If you made it through this far, you've performed differential expression analysis, and keep tuned for videos later down the line that can help with more advanced analyses, particularly with RNA-seq as well as others. Well, thanks for your time.